So I'd like to introduce our next speaker. Professor Gabriele Kaminski-Schierle is the professor of molecular neuroscience at the University of Cambridge, and she's leader of the molecular neuroscience group. Her research revolves around the application of optical techniques to investigate the fundamental molecular mechanisms underlying neurodegenerative diseases, and this includes Alzheimer's and Parkinson's disease. And I shall turn it over to Gabby to tell us about exogenous tau, how exogenous tau affects intracellular, organelle, morphology, and neural signaling.
Thank you so much, Janet, for this kind introduction and for giving me the opportunity to talk today here. I'm really excited. So, okay, so I'm going down in scale again. We started off with single molecules. We then went on all the way to the patients, and now I'm going down a little bit again to the more cellular level. So let me start with a brief outline of my talk. I probably don't need to do an introduction into Alzheimer's disease. I put it there because for completeness. I will then talk about the current intracellular model of tau, especially exogenous tau. Then I will be talking about lysosomal clustering, whether that is a common phenomenon in neurodegeneration, as it has been observed in other neurodegenerative diseases. And then what are the consequences of this lysosomal clustering? I will then talk about the effect of this exogenous tau on lysosomal clustering, as well as other organelles such as the tubular endoplasmic reticulum and the microtubules. And then I will continue by showing how this organelle dysfunction that we can see in these cells is coupled to a synaptic dysfunction as well. And at the very end of my talk, I will be talking about some new tools that we have developed in the lab. These are transparent microelectrode arrays. With those, we might be able to shed light on some of these synaptic defects that we see in the cells. Okay, let me start. I think I can skip that slide as I was saying. I mean, it's really very important to do this kind of research and also to do fundamental research, trying to understand what are the mechanisms that are, the molecular mechanisms that lead to these diseases. So what is the current model of the intracellular fate of exogenous tau? We all know that there is a correlation with the levels of tau in the cerebrospinal fluid and the stage these Alzheimer's patients are in clinically. And so in order to model this, we used exogenous tau, and we added to the cells. And then we studied what happens to this exogenous tau that we added to the cells. We know it is endocytosed and goes into the endolysosomal pathway, and then it can have several different fates. We have shown that it can nucleate in these vesicular structures and lead to aggregation inside, and this can then be potentially, it can be degraded in the lysosomes because it ends up in the lysosomes. But it can also lead to the vesicular leakage, and then it can start to attack the endogenous tau and lead to seeding of endogenous tau. And it can be released, which has been shown by many people in the field, and it can lead to tau propagation. Okay, so we then wanted to study this a little bit more, especially in more detail, using high-resolution imaging techniques. And one of the techniques that I'm going to be talking about a lot today is structured illumination microscopy. Structured illumination microscopy is doubling the resolution. So we can see, this is very good for organelle imaging because we can see structures such as lysosomes, the ER, etc. We can image this very well, and we have, because they are the right size. So if you think of doubling the resolution, something that has a 100 nanometer size is really easily visible using structured illumination microscopy. The other advantage is that you can use it with any dye, and it's also extremely fast. So we used this, and what you can see here, so we used maybe not the correct model because we used COS7 cells. And the reason we used COS7 cells here at the start is because they are extremely flat, and you can see the organelles beautifully. So let me start with what we see first in COS7 cells, and then I will go on to the neurons. So if you look at these images here, you can see beautiful mitochondria, and you can see here in magenta, and you can see the lysosomes in yellow here. And these are control cells. You can see the lysosomes being relatively small as you look at them in these COS7 cells. When we start treating them with 1 micromolar concentration of extracellular tau, we can see that the lysosomes start to cluster in this perinuclear area, and they cluster significantly and also start to swell up. And they are co-localized. You see, this is a lysosomal marker. This is the tau, the labeled, partially labeled tau that we added to the cells. So only 2.5% labeled tau, and then we can see that the two of them are colocalizing. And we can see that this leads to increase in lysosomal biogenesis because the number of lysosomes are significantly increased, and also the area is significantly increased. So there is a swelling happening. Okay, so we thought that was really surprising to see this, because in a previous study that we did, not directly related, this is a model of amyotrophic lateral sclerosis using FUS overexpression. And in this model, using the same type of cells, we have seen that using different variants of FUS overexpressed in these cells, and especially the mutant P525L, more than the R495X mutation of FUS, can lead to increased lysosomal size, but also increased lysosomal biogenesis, similar to what has been observed with tau. And as a positive control here, we used chloroquine, because we knew chloroquine would have this kind of effect. Okay, so what we also observed that this increased clustering of these amyloid proteins in these lysosomes lead to the faulty acidification of these lysosomes. So the pH of these lysosomes is significantly increased. And we again used the same mutant, and we used a fluorescence lifetime technique where we had organ dextran, which is a pH sensor that you can use in cells, and we looked at the fluorescence lifetime change of this organ dextran green that we added to the cells. This organ green dextran accumulates in the lysosomes, and when the pH changes, you can see the lifetime significantly changing. And what you can see here, if you look at the pH here, and this is correlated with the change in the fluorescence lifetime, we can see that again in the P525L FUS mutant, there is a significant increase in the pH. Obviously, the highest increase is seen with chloroquine treatment, which is known to increase the pH in the lysosomes. And we have seen this before. This was a study that was published in 2022 in Nature Neuroscience by the group of Ralph Nixon. And they showed that faulty lysosome acidification in Alzheimer's disease mouse models induces autophagic buildup of A-beta neurons yielding senile plaques. And they call these very nice lysosomal clusters that they see occurring around the perinuclear area, pantos, from the Greek word as meaning flower. Okay, so I'm taking a little step back here. So what we have previously shown in a study published recently in Science Advances, that if the lysosomes or the lysosomes play a really important role in distributing the tubular ER to the periphery of the cells. So what is happening here? So what we did some organelle optogenetics, and we coupled the lysosomes, which you can see here labeled in green. We coupled them to an islet, which is an optogenetically controlled mechanism to couple the lysosomes directly to the motor proteins. And with that, we can force the lysosomes to either go to the cell periphery or bring them back again, depending on which motor we are choosing. And what you can see here, if you look at, especially in magenta, at the tubular ER, if I try to play this here, you can see how the lysosomes, as they are moving to the periphery of the cell, the tubular ER is moving with it. And on this slide here, on the right hand side, we used a similar imaging technique to show that in retinal ganglia cells, that in the axon, which only consists of tubular ER, the lysosomes are important to fill some of these gaps again that you see in the tubular ER. And so you can I don't know whether you managed to see it, that the lysosome managed to fill this gap again. So why is this important? So if you think of the tubular ER being the only structure in the axons, so all the nutrient supplies, for example, calcium, lipids, proteins, to reach the synapse, they are delivered through the tubular endoplasmic reticulum. So if there are gaps, significant, or if there is a retraction of the tubular ER in the axons, you might have a loss of trophic supply to the synapse. And this is what we wanted to study. So now I'm coming back to, after this little excursion, I'm coming back to my story on tau. So we wanted to study this in more detail. And so what we did, we incubated again with COS7 cells with exogenous tau, and then we looked at the tubular ER distribution. And what you can see here, so these are some control images where you see this beautiful tubular ER network in these COS7 cells. And then we have recently published sort of a method to analyze the distribution of the tubular and cheap ER using a machine learning-based segmentation method. And with using this method, we can show the level of, by segmenting this, we can really look at the level or the percentage of the tubular ER in the periphery, and then compare that to the level of sheath ER that we see. If you look at here the tau-treated cells, you can see here that the tubular ER network is significantly destroyed, and the complexity also of the network, by looking at these nodes and edges here using these algorithms, is significantly reduced after tau treatment. And you can see the quantification here that indeed the tubule fraction is significantly reduced, whereas the sheath formation is significantly increased. Obviously, is this important for neurons? And I was saying it's particularly important for axons, because axons only consist of tubular ER. So we did this study here in control cells. You can see beautiful continuous tubular ER in the axons, which is significantly disrupted after tau treatment. Which you can see the quantification of it, you can see on the right-hand side, really indicating that there might be a problem here. To test this, that this is really tau-related, we used a recently discovered small chaperone, which is, has been shown previously to prevent tau uptake by competing with the receptor that is important for tau uptake. And by treating these cells, we can see that this allows us to rescue some of these tau-related endoplasmic reticulum defects that we see in those cells. Okay. We then, because the ER and the lysosomes are directly coupled to the microtubules as well, we also wondered whether the microtubules would be affected. And we can see here, these are again images, high-resolution images of tubular structures in these COS7 cells. And then when we treat them with tau, you can see, I mean, there are fractures in the microtubules. You can see strong bundling. So, and if you look at the intensity change, we can see there is an effect on the microtubules as well. We then, through a collaboration with Ulrich Olsen's group and one of the students in my group, we started to look whether tau actually, this very low dose of tau has an effect on synaptic activity. And Tanya in Ulrich's group, she did some electrophysiology studies here using patch clamping, and we use these YFP channel rhodopsin expressing cells, and we added the exogenous tau, and then we did whole patch clamp recordings in the channel rhodopsin negative cells, and we used the channel rhodopsin cells to optogenetically stimulate those neurons, and then we measured the response. And what you can see here is quite interesting, because as I said, this is only 1 micromolar tau in hippocampal neurons, and we didn't expect it to have really much of an effect. And we're really surprised to see that if you look at the effect of tau, tau itself has hardly an effect, or we can't really see. Obviously, the current is increasing upon stimulation, but it's not affected, the spontaneous activity is not affected in comparison to the control. But if you look at stimulated versus light-induced in the presence of tau, we can see that there is a really loss in the excitatory postsynaptic currents in these primary hippocampal neurons, and you can see some of these traces here, which is quite significant. So we're trying to understand this better, and in order to look at this in more detail, we developed some new fully transparent microelectrode arrays. So current microelectrode arrays using metals such as indium tin oxide, which is not transparent. You can't really see the neurons sitting on these electrodes. And together with people at the engineering department, we developed novel polymer-based and also graphene-based microelectrode arrays. Advantages really that you can do high-resolution imaging through them. So let me just show, this is the preliminary data we've got. This is really early days because we haven't done all the repeats. It's getting obviously the correct device and everything working properly. It's a bit tedious, but we have some indication, especially if we look at the local network and the global network, which is something that you can really show here using these microelectrode arrays. There is a trend that there is a disturbance when we treat the cells with tau and stimulation, but not just by the treatment of tau itself. So confirming the data that we see with the patch clamp analysis. But what the real advantage is that we can really look at these neurons with a very high resolution. You can see here how the calcium, for example, is pumped across these neuronal structures, and then you can correlate these signals. So you have the electrical signals here, you have the calcium signals here at the bottom, but you can also see, for example, if you look at individual synapses here, how these synapses start undergoing structural changes during these calcium spikes. So these are really early days, but we're excited about this and what this can do to dementia research. With that, I come to my summary. I talked about lysosomal clustering. Maybe this is common in neurodegeneration. We can see that tau, but also FUS-induced lysosomal clustering affects the distribution of the tubular R. I didn't show it for FUS, but we do see it as well. And the optogenic stimulation and tau treatment leads to the loss, to the inhibition of the EPSP response in these hippocampal neurons, really indicating that there is some kind of synaptic dysfunction. And we have these new tools to study this. And with that, I'd just like to thank you for your attention and all the people who contributed to that.